Hello. So today I will talk about a way to assess the usefulness of artificial genomes using local ancestry inference method. But first, I will give some motivation. Why artificial genomes? So you are all aware that there is a big surge of genomic data that is caused by next sequencing technologies. But at the same time, there's an increase in the number of privacy laws that restrict the accessibility to sensitive information such as genetic data. And so there is this paradoxical situation where there is a lot of data, but it's not they are not easily accessible. And so most of these genomic data are held by private companies and institutions. And so, for example, there is this famous big biotech company that claims to have more than 14 million samples, but they do not share it with the world. The second motivation is that there are public biobanks, but they are biased towards the overrepresentation of certain population. So they are mostly populated with European samples. And once we have these artificial genomes, we would like to develop some quality metric, some metrics to assess their quality, and also consider some application to evaluate their utility using downstream population genetics task. And so the long-term goal of this project is to view artificial genomes as publicly available and privacy-preserving alternatives to real biobanks. So now I will present the data that we have at hand. So it consists of a SNPs matrix. SNP stands for single nucleotide polymorphism. And so you have the DNA sequences of a bunch of individuals. You have the DNA sequence of a reference genome. And at a given position, you look whether the nucleotide is the same or not. If it's the same, you put a 0, otherwise you put a 1. So you end up with binary sequences that you stack together into a matrix. And this matrix is going to represent the sequence variation between individuals. And that's the information that we are considering here. So more specifically, the data is coming from the 1000 Genome Project, and we have 2,500 phased haplotypes, which correspond to the individuals. And these haplotypes are containing 65,000 SNPs, covering a region of 80 million base pairs in the chromosome 1. And so if you do, for example, a PCA on this data, you can see that it captures population structure, where the modes correspond to the different continental ancestry groups, and in particular, to this continental ancestry group correspond different SNPs pattern. And we have also more detailed information about the population within each superpopulations. So we have, for example, the labels of Japanese, Mexican, Peruvians, etc. And so I will give a brief plan for the next minute. So I will talk about generative modeling in general, but this is not the focus of my talk. Then I will give some application on how we can use them. I will move on to the particular application that we chose, which is ancestry inference. And then I will give some results on this task, which is going to be a classification task. And the overall objective of this presentation is that I will compare artificial genomes coming from different generative models and test them on this downstream population genetics task. So there are two families of generative models, the data-driven one and the model-based one. So the data-driven one are deep learning-based. So you have, for example, generative adversarial network or restricted Boltzmann machines. So these are for so these were tried by the Heraklion in 2021 and 2023 to generate genomic data. And you also have autoencoders, diffusion models, language models. And in the model-based one, you have coalescent and forward simulators. And you also have some simulation based on the chunking of haplotypes and shuffling them together. And so we chose the diffusion model to generate our genomic data. And so I will very briefly explain the process of the diffusion model. So you have the forward process, which is a noising phase, where you have your original data, and you are going to add noise to it. And to train such a network, you will sample a random time step, and to these time steps correspond a certain noise level that you are going to inject into your original sample to have a corrupted sample. And so your neural network is going to take as input the corrupted data and is going to denoise it. And so concretely, it's going to predict the noise that was inside the noisy sample. Once you have your neural network that is trained, you can just sample some random noise and denoise it bit by bit by going through the whole chain. And so we use this model to generate artificial genomes. So we generated 65,000 SNPs using this diffusion model, particular diffusion model, that was also conditioned on the superpopulation labels. Now I'll move on to some applications for this synthetic data. So we could do some selection scan or imputation. Both were tried in a Boak-Hellman paper in 2021. So in selection scan, you are interested in detecting signatures of selection. In imputation, you have some corrupted data and you want to recover the information at specific sites. And we could also envision an application for ancestry inference. So this was done in a Montserrat paper. And in progression detection, where we would like for this task to either augment the real reference panel with this synthetic data, or completely substitute the real reference panel with the synthetic data. And so we chose the ancestry inference task because it was already done, but we dig this further. So in ancestry inference, you can do global or local predictions. So the genetic material of individual is inherited by ancestors that come from different continents. And each individual in a population might have different ancestry coefficients. And so here, each color represents these different sources population, sources ancestral population. And if we dig further, we can see that these ancestries are going to be spread along the genome. So we can do actually some prediction. We can predict the ancestry of a particular region along the genome. And there exist multiple methods that does that. So there is RFMix, Linet, or Genomics, for example. And here we chose Linet because we saw that it gave similar performances as the other, but was faster. And so how does Linet work? It takes as input phased haplotypes. And it's going to chunk these haplotypes into 500 SNPs segments. And there is as many neural networks as there is segments, 500 SNP segments. And so each network is a fully connected architecture that is going to take 500 SNP segments and will output a vector of probability for each of the sources population. And at the end, we can, by taking the argmax operation, we can predict the ancestry of a given segment. And there is an implicit step that is made by the Linet algorithm, which is that it's going to simulate recombination for a certain number of generations. So you start with this supposedly unadmixed or the least admixed haplotypes, which are chosen from the sources population. And it's going to shuffle them to simulate the recombination. So what actually is what's seen by LineNet is this we combine these admixed haplotypes to a certain extent. So now I will present the setup of the experiments. So to train a LineNet model, we have the real data and the synthetic data. So we chose for the both data, they should contain European, African, and East Asian samples because they are the least admixed population that we have. And then the synthetic data is coming from two generative models. So we have the diffusion model and also a baseline, which is composed of three Bernoullis, where the parameters of each Bernoullis are computed from the allelic frequencies of the samples coming from the superpopulation. So we have three Bernoullis with three different parameters. And so we train multiple linets, so a linet on the real data and a linet for two linets for both synthetic data. And then once these models are trained, we can do ancestry inference and use the held out test set that is composed of this three sources population, and also another test set from the real data, which are composed of American samples, because these are already admixed. And so in particular, for the first test set, we constructed a ground truth by using the AppTools algorithm. So it's going to take as input these haplotypes from the sources population, and also a set of parameters like the ancestry coefficients, which correspond to the proportion of ancestry that we want along the output haplotype. And it also takes as input the number of generations for which the simulations are going to take place. So at the end, we exactly know the ancestry for each position. And so here are some results on the ground truth data. So we train, here we have the LineNet model that was trained on the real data. And I plotted the accuracy of this model. So it's pretty good overall. It's over 90% up to 96%. Then we have the LineNet model that was trained on the synthetic data coming from the Bernoullis. And here, surprisingly, it's already decently good from 77% up to 88%. And finally, we have the synthetic data coming from the diffusion model. So it's the LineNet model trained on this data. And here, the performance kind of matches the model trained on the real data while being slightly lower. And we have a second experiment. So we have the second experiment on the test set consisting of American samples. And here we do not have the ground truth. So what we did is take the LineNet model trained on the real data, take the one that is trained on the synthetic data coming from the diffusion model, and you make the prediction for each of these haplotypes, and you align the predictions. So we computed a percentage of identity between both predictions. And we can see that for both models, the alignment is pretty good. And it's less good for the model trained on the real data and the Bernoulli, the data coming from the Bernoulli model. And in particular, we can go in more detail about these numbers. We can plot the proportion of predicted ancestry for each population within the American continents. And so we can also show the percentage of identity for this population. And we can see that it's lowest for the Mexican and the Peruvians. And also that the model trained on the synthetic data is doing more predictions for the East Asians. And we did also another experiment where we increased the size of the synthetic data. Now it's 4 times bigger than the real data. And we see that it's increasing quite a lot the performance of the model trained on the Bernoulli data. So now I will move on to the last experiment, which is the deep generative ensemble approach. In this scenario, we have a data holder that has the real data, but that do not want to share it with the world. And we have a data user that would like to use this data to solve a given task. So to please everyone in this world, the data holder could naively train a generative model on this real data and create a synthetic data set. And that would be released to the world. And so the user would train his machine learning model on this synthetic data. Or you have the deep generative ensemble approach, where the data holder would train multiple generative models, create multiple synthetic data sets. Those would be released to the world, and the data user would train multiple machine learning tasks on each of these models. And so we reproduce these experiments where, so we had 10 runs, and in each run, we trained K generative model, created K synthetic data sets, and trained K lineages. K was taken to be equal to 5 here. And so here we have the results for the real data, which are exactly the same as before. And we have then for the naive setup, where we train we trained 10 generative models, created 10 synthetic data sets, train K machine learning model on this, and make the predictions for each of these models. And so here we can see that it's matching the performance of the real data while being slightly lower if you look at the Y scale. And then we have the naive big approach, where the synthetic data set is 4 times bigger than the naive approach. And here we can see that it's increasing the performance of the predictor, but it's also risky because you are putting a lot of trust in your generative model. So if it's overfitting, it might cause privacy leak events. And finally, we have the deep generative ensemble scenario. And we can see that it's increasing with respect to the naive approach, while being it's a very slight increase in performance. And it's supposed to be more robust to some generative uncertainty in the generative model. And so I will conclude by recapitulating. So we train a diffusion model to generate some artificial genomes condition on the superpopulation labels. And then we benchmark these generated genomes using ancestry inference. And so we saw that the ancestry inference method was performing similarly when it was trained on the real data and the synthetic data. So we can say that the information content is equivalent between artificial genomes and real data. And so that from the quality of the predictor, we can say that these artificial genomes are indeed useful for this task. And then we saw that the deep generative ensemble approach improved the model training, and it could be more useful also in a scenario where the data user would not have access to any real data, but only synthetic data. And that would alleviate some the bias that this would cause. And finally, I would like to conclude with this word that would like to view artificial genomes as publicly available and privacy-preserving alternatives to real biobanks. Thank you.